Good afternoon, everyone. Welcome back to those of you who have been here for much of the day in a really wonderful symposium on mitochondrial biology and disease. And welcome to those of you who haven't been here during the day and who've come now for the Wednesday afternoon lecture to be delivered by Douglas Wallace. The title being A Mitochondrial Etiology of Metabolic and Degenerative Diseases, Cancer and Aging. Sounds like he's going to cover a lot of ground here, people, so hang on to your hat. I've had the pleasure of knowing Doug for about 30 years, since we have shared interests in the field of human genetics. He got his undergraduate degree at Cornell. He spent 2 years in the public health service. Was that here? No, I was hoping maybe you were in the NIH gang of the Yellow Berets in a particular era there, but doing something. And then went on to get his PhD in microbiology and human genetics at Yale. After that, he spent 7 years at Stanford. I guess he's just one of those people who gets restless after a while, ended up at Emory, where he was for quite a significant period of time and a very productive period of time, almost 19 years. Then moved to University of California, Irvine, and most recently, 4 years ago, has moved to the University of Pennsylvania and Children's Hospital of Philadelphia, where he has perhaps one of the longest titles I've seen in a while, the Michael and Charles Barnett Endowed Chair in Pediatric Mitochondrial Medicine and Metabolic Disease. Do you have to say that when you introduce your? I hope not. Anyway, he has more than anyone else really defined the field of human mitochondrial genetics, identifying the method of inheritance of mitochondrial DNA back there quite a few years ago, demonstrating the very first example of a disease that is inherited because of mutations in mitochondrial DNA and therefore inherited through the maternal line, and a long list of other achievements for which he's been recognized by election to the National Academy of Sciences as well as the Institute of Medicine, and most recently received the highest prize that is given in genetics, the Gruber Foundation Genetics Prize in 2012. So we're very fortunate to have him here to talk to us this afternoon. Please join me in welcoming Dr. Doug Wallace.
So I want to really thank you, Francis, for being here and for that gracious introduction. Thank you very much, Steve Zulo, for all the effort you put into having the fabulous speakers we've heard today, and for all of you for being here this afternoon. We are at, as Francis just mentioned to me, a unique time. There's no time in Western medicine, biomedical science, when we have more tools and more opportunity to make real impact on the health of our society. At the same time, we're looking at continual decline in the support for our sciences, with particular anxiety and concern for the future of our young scientists, who, of course, are the future for this field. And we have to ask ourselves, I think, at this point, why is it that our society is turning away from the concept that biomedical research could actually be a major asset to them? Why are they then pulling back from their support? And I think the reason for that is that, unfortunately, there is a inverse correlation between the frequency of the diseases that impact our society and our ability to diagnose them effectively and to treat them. And this means that for the vast majority of the American citizens, and I think also for those throughout the world, there's a growing disillusionment with what medicine can do, since these are the very problems that they are confronted with. So having this problem in front of us, which I think we must admit is true, we have to ask ourselves then, why is it, with all the effort that you all are putting in and with all the support that governments and private industry has provided throughout the world, why haven't we been able to do better? Why haven't we cured a single case of Alzheimer's disease? Why is diabetes increasing? Why is obesity an epidemic, et cetera, et cetera? And there was a philosopher of science called Thomas Kuhn, and he proposed a number of years ago that when scientific effort is invested more and more heavily in trying to solve some problems, and yet progress seems to not be moving forward at the rate you would expect, that it may be time to ask what are the basic premises on which these endeavors are being addressed, and are those in fact the right ones for addressing these problems. And what are the problems that we are concerned about? Well, of course, there are all the neuropsychiatric diseases. Being in pediatrics, and I went to CHOP for a specific reason to look at pediatric diseases, but one of the most obvious is autism, which is now assumed to be 1 in 88 boys being affected. Or Alzheimer's, or Parkinson's, or migraine, or depression, or schizophrenia, or obsessive compulsive disorders, or in the heart and muscle, cardiomyopathy, cardiovascular disease, myalgia, chronic fatigue syndrome, or the visceral diseases, renal, hepatic, and gastrointestinal diseases, or metabolic diseases, type 2 diabetes, obesity, hypertension, inflammatory diseases, type 1 diabetes, multiple sclerosis, lupus, cancer, and aging. All of these are, in fact, the major concerns we have, and yet they are the ones that we seem to be least able to address. So if, in fact, Kuhn is right, that maybe there's something wrong with how we're approaching these problems, we should then ask, well, what are the basic assumptions on which we're approaching these problems? And I'd like to propose that Western medical philosophy has been based on two basic ideas. One that goes back to a man named Vesalius, who lived about 500 years ago in Italy, and he was the first to define the anatomy of the human body. And that was a tremendous breakthrough because that then began to systematize medicine, and of course then all future physicians and future investigators then began to specialize in different parts of the body. So now today we have ophthalmologists and neurologists, cardiologists, nephrologists, all organ-specific specialties. Well, that's all fine and good, but it led to an unspoken corollary. And that unspoken corollary is if you have a headache, then you get referred to the neurologist because the assumption is there's something wrong with your head. That is, the idea is if you have a specific tissue-specific symptom, this must be due to a tissue-specific defect. Then the other idea, which goes back to Gregor Mendel, who was studying the inheritance of pea plants, he observed that certain subsets of the hereditary factors followed a pattern where it seemed like the adult plant had 2 copies of something. Each sex cell got one of those copies, and then the new offspring got 2 copies again. And we now call that the laws of Mendelian inheritance. And then in the turn of the century, the Drosophila geneticists who were studying the anatomy of the fruit fly realized that almost every one of their anatomical problems was inherited according to that pattern, and they soon learned that the chromosomes followed those same ideas, and that led to what we call the Mendelian laws of inheritance. Now, the idea that there is a law or laws of inheritance also has an unstated corollary, and that corollary is if those are the laws of inheritance, then if something is transmitted in a family according to the laws of Mendel, it's genetic, and if it's not, then it's environmental. So these two ideas have held us in good stead. We've made tremendous progress, but maybe they've exhausted some of their capacity to serve us. Because, in fact, all of the Mendelian genes on chromosomes are also where all the anatomical genes are. So there was an internal consistency. An anatomical change would clearly be Mendelian. But life is not just about anatomy. It's also about being animated. And in fact, you're the most animated thing in our known environment. And Newton indicated, again, half a millennia ago, that any kind of matter was inanimate unless it was acted on by energy. And only energy animated matter. So, in fact, the fact that you're highly animated means that one of the most important things about you is the flow of energy. So, therefore, we can realize that life is really the interaction of anatomy, energy, and the information for anatomy and the information for energy. So once we realize that energy is something we should also be looking at, then we can realize that different organs rely on energy to different extents. So you could then imagine a situation where you could have a partial energetic defect, and it would specifically affect those organs that had the highest energy demand. And that, in fact, turns out to be the brain, heart, muscle, renal, and endocrine systems. And then you could also come to realize that, in fact, some of the most important energy genes are not inherited in the chromosomes and therefore not according to the laws of Mendel. And therefore, this whole aspect of inheritance had been overlooked from a clinical point of view. So now we really have basically 4 sets of paradigms, and when we add these together, I hope to be able to convince you we'll have a more coherent view of the disease process. So where did this dichotomy of energy and anatomy come from? Well, it came from a symbiosis that occurred over 2 billion years ago between a methanogen, which was an archaeobacteria, and an oxidative bacteria, which was of what's called the alpha proteobacteria, and it was more oxidative. And these 2 organisms came together because of the changing environment to form a relationship. And that relationship ultimately became tight enough that they merged together. And the methanogen's genome, which was the same size as the original bacteria, began to accumulate genes because bacteria exchange genes. That's what they do. And so there was a gene flow from the oxidative bacteria to the methanogen nucleus. And you might ask, well, why would that be? Why would there be a gene flow? Well, it turns out that it takes about 1 bacteria-sized cell's energy to replicate its DNA, transcribe it into RNA, and translate it into protein. And the reason that bacteria don't have bigger genomes is they can't make enough energy. So energy is limiting through the bioenergetic process, through the biosynthetic process. So, in fact, if all the bacteria maintained their full genome, they would use up all the energy, and there would be no advantage. But if, in fact, a gene from the bacterium was transferred to the nucleus, then instead of having to have 1,000 copies, 1 in every bacterium, you could have only 2. Now, those 2 in the nucleus could be replicated, transcribed, and translated with 1,000-fold savings of energy. So by this transfer, then, of genes from the mitochondria to the nucleus, there was a tremendous energy savings. And that energy savings, then, created enough raw energy to allow the nucleus to accumulate 25,000 genes and thus allow multicellularity to occur, formation of arms, legs, thought, and questions. Okay, so then what we really have, then, are 2 organisms that have undergone a specialization. The nuclear cytosol has become specialized in anatomy, and the mitochondrial has become specialized in energy, or the vital force. Now, how does this energy flow through our cells? Well, for us that live on the surface of the Earth, high-energy photons impinge on a cyanobacteria, which is a symbiont in plants, giving chloroplasts. And the cyanobacteria takes the energy from the sunlight and splits water into hydrogen and oxygen, releases the oxygen to the atmosphere, and then the plant then uses that hydrogen, condenses it with carbon from CO2 to give you sugar. And the reason, in fact, that we've cleared off the entire Midwest and grow wheat is because we get the energy of sunlight from the sugar in the starch in those plants. So then we eat the starch goes into our cells, it's split into 2 parts, pyruvate, they enter another symbiotic bacterium, the mitochondria, that strips the hydrogens off the hydrocarbons, react it with the oxygen that we're breathing to give you the water back, and release the energy in chemical form as ATP and in kinetic form as heat. So the important point here is that all energy flow is not through the nuclear cytosolic organism, but through symbiotic bacteria. And so until we understand the flow of energy through the symbiotic bacteria, we can't fully understand the pathophysiology of complex disease. So this is a mitochondrial biochemistry lesson from hell. I've never had a medical student that liked it, but in fact, I love it, and therefore you'll be tortured. Here we have then an outer membrane, an inner membrane space, an inner membrane, and a matrix. This is the mitochondria. Glucose has gone through glycolysis to give you pyruvate. If the mitochondria is defective, the pyruvate can be reduced to give you lactate, or an amino group to give you alanine. The pyruvate goes through pyruvate dehydrogenase to give you acetyl-CoA, and that drives the tricarboxylic acid cycle, the purpose of which is to strip the hydrogens back off the carbon and put them into this carrier, NAD, to give you the reduced form NADH. Now we're going to burn those hydrogens with the oxygen that you're currently breathing, and the electrons flow down this wire, complex 1, CoQ, complex 3, cytochrome C, complex 4, reduce an atom of oxygen to a molecule of water. Now, the problem with electron flow, like any electrical system, is it's very difficult to store that energy because it's kinetic. So how, in fact, did Mother Nature solve that problem? She solved that problem by creating a capacitor. So as the electrons move through complex 1, 3, and 4, it pumps positive charges through the insulator of the inner membrane into the intermembrane space to give you acid and positive on the outside and alkaline and negative on the inside. And this was the great insight of Peter Mitchell. then this stored energy can then be used for lots of purposes, one of which can be used to convert low-energy ADP into high-energy ATP, which can then be exchanged by the adenine nucleotide translocator. The ATP of the ADP goes out through the voltage-dependent anion channel, and therefore the ATP can do work. So, in fact, we've coupled oxidation with phosphorylation, and hence oxidative phosphorylation, or OXPHOS. Now, it turns out that everybody in this room has a different efficiency at OXPHOS. Some of you are very efficient at burning the hydrogen to pump protons out and converting the hydrogen into ATP, and we would call that tightly coupled. And since every calorie that you eat is 1 unit of heat, the most efficient people eat the least calories for the maximum amount of work and generate the least amount of heat. On the other hand, other people are less efficient in pumping protons out and in converting them to ATP, and these people are loosely coupled, and therefore, to make the same amount of ATP, they have to eat more calories. So by burning more calories, they're making proportionally more heat per unit work. So this coupling efficiency, as I'm going to try and argue, is a very, very important component of the variation of people in this room and in the world. Now, this system is like any furnace, and it has incomplete oxidation, combustion, and that is personified by the electrons in complex 1 and 3 being able to donate directly to O2 to give you superoxide anion. With an unpaired electron, which wants to oxidize lipids, proteins, and DNA, we have an enzyme manganese SOD that will take 2 of these and dismutate it into the more stable hydrogen peroxide. But as you heard earlier today, in the presence of transition metal, you can get another electron to give you hydroxyl radical, which has also an unpaired electron, and these then will oxidize your mitochondria and your cells, and they are the so-called oxygen radicals. But you'll also see they're very critical in signaling. The mitochondria, being a capacitor, is also very good at picking up positive charge, and it has a selective system for regulating calcium. And it also has this system called the permeability transition pore, which is a self-destruct system. Normally, it's a closed door, maintaining the membrane potential. However, when the membrane potential declines, high-energy phosphates decline, calcium overload becomes high, or oxidative stress is high. They all impinge on this structure, goes from an closed door to an open channel, shorts the circuit, the membrane potential, the high osmotic strength of the matrix causes fluids to flow in, intermembrane swells, backs and back form a megachannel, they release these stored pro-apoptotic proteins, they go out and degrade the cytosol and the nucleus. So the cell self-destructs by degrading from inside out when mitochondrial energetics is impaired. So the mitochondria then generates much of the energy, regulates redox, as we heard today, generates reactive oxygen species, which are signaling molecules, but at high levels become damaging, regulates calcium, regulates cell death, and is central to all intermediate metabolism, including regulating things like acetyl-CoA, ATP, and S-adenosylmethionine. So basically then, what we can think of the cell as 2 life forms: a nuclear cytosolic life form whose genes are located in the nucleus, transcribed in a message, read on cytosolic ribosomes that are sensitive to cyclohexamide and emetine, and about 1 to 2,000 of the nuclear encoded proteins were originally from the mitochondrial DNA and are now made on the cytosol and vectorially transported to make the anatomy of the mitochondria. But the mitochondrial DNA is 16,569 base pairs, codes for small tRNAs, small and large ribosomal RNA, 13 polypeptides. These are translated on mitochondrial-specific ribosomes that are initiated by informal methionine, just like any bacterial ribosome, sensitive to chloramphenicol and the metoglycosides. And these 13 proteins are 7 of the 45 proteins of complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5. So you might ask, if you put 2,000 genes in the nucleus, why keep 13? And the answer, I believe, comes from the fact that every one of the proteins is either complex 1, 3, 4, and 5. And what do 1, 3, 4, and 5 have in common? They have the membrane potential. So these proteins then are the wiring diagram of the power plant. And if any one of these complexes became leaky for protons, it would short the capacitor. And once the capacitor collapses, in fact, you lose your potential energy, and then you stop breathing, and then you become inert. We call that dead. Okay? So this then is critical that this system be maintained. All of these enzymes have to co-evolve and be balanced together. So how does that occur? Well, the problem then is you can't have recombination, because if we mixed and matched mitochondrial DNAs from any two of you and you had slightly different circuit diagrams, then we get a mismatched circuit and it would short. So in fact, we cannot allow recombination, but that's what the nucleus does. So Mother Nature had to solve this problem. Being a woman, she solved it in a very logical way. She said that the mitochondrial DNA would be inherited only from the mother. So the mitochondrial DNA is inherited from mother to all of her children and her daughters to their children, whereas male's mitochondrial DNAs enter the egg, are seen and foreign, and are selectively thrown out and destroyed. So men have been thrown out for 2 billion years. Don't feel bad. Now, each cell then has thousands of these mitochondrial DNAs, and they're constantly replicating inside your cell. So they're replicating, they're increasing in number, but they're being eaten up by what's called mitophagy. So your colonies are in a steady state. But since they're replicating, they accumulate mutations, and these mutations then create a mixture of mutant and normal bacteria inside your cell, and we call that heteroplasmy. So red would be mutant, blue would be normal. If the cell divided down this way, both cells would have some mutant and normal, but if it divided this way, then that cell would have only normal, and this would have twice as many mutant. So the point of this is that mitochondrial DNA genotypes segregate during mitosis. So a heteroplasmic zygote, let's say with the Leber's mutation, will give rise to twins with different mitochondrial DNA genotypes and with totally different clinical phenotypes. Or if it was a heteroplasmic mutation and went through many divisions, then you could have some cells that would segregate to primarily normal mitochondrial DNAs and others primarily mutant mitochondrial DNAs. And the more mutant mitochondrial DNAs there were, the less energy, and it would be the equivalent of metropolitan brownout. As the energy declines, then those organ systems with the highest energy would be the first to begin to malfunction, and that turns out, as you'll see, is the brain. So we call that the bioenergetic threshold. And we have then a very high mutation rate, and we'll talk a lot about why that is, but the mutagen is probably ROS. So the mitochondrial DNA then codes for a small and a large ribosomal RNA, 22 transfer RNAs that punctuate the genes, ND1, 2, 3, 4, 4L, 4, 5, and 6 for complex 1, cytochrome B for complex 3, CO1, 2, and 3 for complex 4, ATPase 6 and 8 for complex 5. And it has a control region that regulates transcription and replication. So it has a very high mutation rate, and one of the things that we get is maternally inherited diseases, new mutations that occur in the female germline. For instance, this mutation in the 12S ribosomal RNA at 1555, which is a nucleotide position, if you inherit that from your mother, you're fine until midlife, and then you'll lose sensory neural hearing loss in your ears and go deaf. Mutation in the tRNA leucine gene at 3243 at a low percentage mutant gives you diabetes, and a high percentage mutant will give you strokes, and an even higher level will kill you as an infant. Mutation in the tRNA glutamine gene is found in high percentage of late onset Alzheimer's and Parkinson's disease. Mutation in the tRNA lysine gene at 8344 will give you a kind of epilepsy. There are literally hundreds of these protein synthesis maternally inherited diseases. But there are also missense mutations, like a mutation in the ND1 gene at 11778. If you inherit that from your mother, you'll be fine until midlife. Then you'll lose vision in one eye, then in the other, and you'll be legally blind, Leber's hereditary optic neuropathy. And up here, ND6, the same mutation, mutation 14484, gives you the same phenotype. Mutation in 14459, which is more severe, when it's heteroplasmic, will give you the optic atrophy. When it's homoplasmic, gives you dystonia. Mutation in the ATPase6 gene at 8993 at 70% gives you retinitis pigmentosa, 80% olivopontocerebellar atrophy, and 90% death by Lee syndrome as a child. These are just some of the cytochrome oxidase subunit 1 genes that we found in various kinds of prostate cancer. We could talk about cancer a lot, but we already heard several very nice lectures in that area, so I've dropped them for time's sake. So the mitochondrial genome then involves thousands of 1 to 2,000 nuclear genes, these 35 mitochondrial genes. We have maternally inherited diseases. But we also have a lot of variation. Everybody in this room probably has a slightly different mitochondrial DNA sequence. And some of these are very common, like this variant, which we call lineage L, or haplogroup L, in the ND1 is found in 3/4 of sub-Saharan Africans. This variant H is found in half of Europeans. Variant A, B, C, and D are found, arose in Asia, and they crossed the Bering Land Bridge and colonized the Americas. And these, I'm going to argue, change the mitochondrial physiology to allow people to adapt to different environments. Okay. So once we begin to think energetically, getting close to the introduction, be patient, Once we begin to think energetically, then we can realize that we can think about anatomy from an energetic point of view. So we have high energy utilized tissues, like the brain has the highest energy demand and the lowest reserve. It's 2% of the body weight and uses 20% of the oxygen. Therefore, a 10% reduction in energetics is going to give you a very bad headache. But we also have effects on heart, muscle, renal, and endocrine. They have constant energy demands, but higher energy reserves. So sometimes they can be more tolerant. We have energy storage tissues. White adipose tissue stores fat to make ATP. Brown adipose tissue stores fat to generate heat. We have an energy homeostasis tissue, the liver. Why the liver, and why does it regulate blood glucose? Because that's your connection with sunlight. And we have an energy sensing tissue. The pancreatic beta cell sees high glucose, sends out the signal insulin, and tells all the cells switch over toward glycolysis, or the alpha cells sense low glucose, sends out glucagon and says, turn on cyclic AMP, initiate oxidative phosphorylation to burn fat. So all of this then leads to this concept that if we now move anatomy out of the center of our thinking and put in energetics, then all of the common complex diseases could be understood by the same pathophysiological mechanism. So the center then would be oxidative phosphorylation, not because glycolysis is unimportant, but because this is the majority of the energy and the most sensitive. So we could have variation in nuclear genes, either mutations or polymorphisms, or changes in their expression, the epigenome. That would have changed energetics. We could have mitochondrial variants, ancient adaptive polymorphisms, recent deleterious mutations. That would affect energetics. But this is how you process all your calories and the oxygen that you're breathing. And it's where the source of energy to grow, mature, and reproduce are. And it's also the most sensitive to toxins. So if you don't like your neighbor and you want her to stop bothering you, just put a little cyanide in her tea. She'll stop bothering you. So almost all the major toxins are, in fact, direct inhibitors of mitochondria. Why? Because that's your Achilles heel. So if you inhibit mitochondrial function, then in fact your mitochondrial replication is perturbed. You will accumulate somatic mutations because replication is not going well, and that will give you a progressive decline in function. And that gives you the delayed onset and progressive course that we see in all complex diseases and aging. And so we believe then that's the aging clock. And what would be the intermediates where you have partial defects? Well, they're going to affect the brain, the heart, the muscle, and the renal, and that gives you all of those complex diseases. But it will also perturb the flow of energy through your system, and that will then give you the metabolic diseases. The mitochondria is the most common bacterium in your body. There are 10 to the 14th bacteria in your gut. There are 10 to the 17th in your cells. So every time a cell breaks open, if the mitochondria are not digested, then you're going to release those N-formal methionine polypeptides and the mitochondrial DNA and cardiolipin and a lot of other things, and you're going to amass a massive inflammation response. And this is a very growing area right now, the so-called DAMPs. The whole inflammation system, we think, is secondary to most of the degenerative diseases. And finally, how you manage energy is critical to whether cancer grows. And it's not that one system or the other is defective, it's that the cancer cell has the ability to switch. Okay, so that's the background. Now we're going to have some examples. So this is just a family where this person went blind, related to this female, to this person went blind, to these people that went blind, to this female, these men went blind, and this woman, and then through this female to this blind person. And then, of course, it's a late onset disease, so the young people didn't show it. Now, this is Leber's hereditary optic neuropathy. Perfectly fine till midlife, suddenly lose vision in one eye and then in the other. And this is a missense mutation in the ND4 gene, arginine codon 340 to histidine. Now, some of the things you can immediately see about this. First of all, not everybody on the maternal lineage is blind. That's why people thought this was an X-linked gene originally. And obviously, there's a 4-to-1 preference in males. Now, that's rather curious. Why would you have a maternally inherited disease with male bias? Well, what you're going to see is male bias is, in fact, a theme. And one of the interesting things about male bias is that you also see it in autism. You also see it in Parkinson's disease. Many of these complex diseases show this male bias, which we think is a sex-limited feature of energetics. But still, that asks the question, why is it that there's variability in expression in a mitochondrial DNA disease gene? And one of the major factors is the background on which that mutation occurs. So the mitochondrial DNA, as I said, almost everybody who has a different mitochondrial DNA. So you can imagine that a Leber's mutation would have different effects on you, and that's exactly what we see. So here are 3 different Leber's mutations. There are now dozens, But this one, 3460, and the ND1 gene causes a severe complex 1 defect, 11778 ND4, a moderate defect, 1454 is a mild defect, yet they all give the same phenotype. Why would that be? The answer is that the background is important. This is haplogroup J, found in 20% of people of European background. So this severe mutation, it doesn't matter your background, whether it's J or not, you're going to go blind. But this more moderate one, 1/3 of the people are also J. So J had to increase the penetrance. And for this very mild one, almost 3/4 are J. So in fact, J increased the penetrance of these milder mutations. Here's another mutation, ND1 103394C, tyrosine codon 30 to histidine, and that also will increase the penetrance of the milder mutations. So what are these lineages? So these lineages are, in fact, continent and regional specific mitochondrial DNA lines. So if I sequence the mitochondrial DNA of everybody in the room, any two of you, if a number of nucleotide substitutions between you would be directly proportional to you shared a common mother, because, in fact, mutations can only occur along the maternal line. So by using that logic and then sequencing the mitochondrial DNA of indigenous people around the world, we found that, in fact, mitochondrial DNA initiated in Africa about 150,000 years ago, gave rise to L1 and L2, which are types of pygmies, L3 sub-Saharan Africans. L3 gave rise in Ethiopia to M and N. M and N moved along Southeast Asia, staying in the tropics all the way down to Australia. But N did something very unique. It moved north into the temperate zone, giving rise to lineages IWX, and then through R to lineages H, J, T, U, UK, and V. And then later, M moved north, acquiring mutations to give you C and D, and N gives rise to A, and A, C, and D became enriched in Chukotka, and 20,000 years ago colonized the Americas. So here's something very strange. Nuclear genetic variation is panmictic. All the alleles are found throughout the world, but mitochondrial DNA variation is highly geographically constrained. So why would that be? Because these women, if they want to reproduce, they need to be able to run away from lions, otherwise they get eaten. So how do they run away from lions? They have to have very efficient oxidative phosphorylation to make a lot of ATP. Therefore, they need to be tightly coupled and make a minimum amount of heat. But up here, the lions froze to death. That wasn't a problem. The problem is they're going to freeze to death. So how are they going to solve that problem? By accumulating mitochondrial DNA mutations that decrease the coupling efficiency. Now they have to eat a high-fat diet for the same amount of ATP, but that's going to generate a lot more internal heat. And they'll have to kill marine mammals, and everybody in Washington will look down on them. But sorry, that's their niche. That's the way it is. So anyway, the point is that these mitochondrial DNA lineages became adaptive, and they allowed our ancestors to live in these different geographical regions. And this just shows out of Africa, the lineage M, which stayed in the tropics, it only has synonymous mutations. Those are random mutations. But N, which moved into the temperate zone, has two missense mutations, ND3 and ATPase6, alanine 114 threonine, and alanine codon 59 threonine. And what does that do? It decreased the mitochondrial membrane potential and altered calcium metabolism, making it less efficient. Every time we have a new branch to the mitochondrial DNA tree, it's founded by an amino acid substitution that affects a highly conserved amino acid. So this variant in T, 4917, in the ND2 gene, this amino acid is conserved all the way down to C. elegans. These mutations in the cytochrome B gene, 14798 and 15257, this one is conserved in all mesozoans. This is conserved all the way to E. coli. Now, wait a minute. What I'm telling you is these amino acid changes that are conserved throughout species evolution are polymorphic in this room. So that is upside down from everything we've been taught about molecular evolution. So how could this be? Well, remember that the mitochondrial DNA has this incredibly high mutation rate. So how would it work then? The idea that we have is that so this is the mean environment. That's the most common environment. So we have then a mitochondrial lineage that has become adapted to an extreme environment with 2 standard deviations from the mean. It then is going to acquire mutations, some of which are going to be functional. That will then create lineages that move back toward the modal environment, ultimately fixing these functional mutations with random variation.
that then will radiate and then acquire these functional mutations and move into another extreme environment, creating then another node, which is then enough able to create a new species. And then you go back through the system again. So when you take the average of the species, the environment has selected for convergent evolution on the same amino acid changes. But at extreme ends, we have then these functional variations that we see in the Arctic or in the extreme Africa. So I think then this is the driving force for adaptation within species, whereas nuclear variation is the driving force for adaptation between species. So here's an example of that. This is studies we did in Tibet. We've sequenced mitochondrial DNA from Tibetans at many altitudes. There is, of course, lineage N and M, just like we saw out of Africa. I did forget to say that out of Africa was M and N. But what we found in Tibet is that this mutation, T3394C, arose 3 independent times. If we now ask, what's the frequency of that mutation versus altitude, we see as the altitude of the village increases, so too does the 3394C. This particular lineage is founded by this mutation, and it, of course, also increases with altitude. So there is an odds ratio that this occurs by chance of over 25. So what this means is this mutation is being selected by altitude. But wait a minute, that's the same variant that we said increases the penetrance of labors. So how could it be bad for labors and good for altitude? Okay. This is another variant. We're going to come back to that. This is just another lineage. This is lineage F4, and this is just looking at obesity. And it's one of many, many studies that we've been doing like this. But you can see that F4 has a significant effect in increased BMI levels. And we can do that for diabetes and cardiovascular disease and on and on. This is another variant. This variant occurred in the tRNA glutamine, an A to G. This arose about 7,000 to 10,000 years ago in Europe. It forms a distinct lineage. It's only about 0.4% of Europeans. But if you now look at people with Alzheimer's and Parkinson's disease, late onset, 3% have Alzheimer's, 3% of Alzheimer's have the mutation, 5% of PD, 7% of ADPD. So it predisposes to late onset neurodegenerative disease. We get a mutation that arises independent of that spontaneously, 3397. This is a methionine codon 31 to valine, but that's the adjacent codon to the Leber's mutation. And this one gives you ADPD. Okay, so how do we make sense of this? What we realize now is that the phenotype of a mitochondrial variant depends a lot on context. So out of Africa, we have L, and that gives rise to N with these 2 founding mutations, and the tRNA glutamine mutation gives this rare form that is predisposed to AD. But we can also get this mutation, 3397, and that now is fairly toxic, and it gives you ADPD rarely. However, you can get the 3394 mutation, tyrosine codon 30 to histidine. That is at a higher frequency, and that increases your risk of labors, but also obesity and diabetes. But over here, when we've skipped these mutations, then in that context, at a high altitude, this particular mutation is now common, and it's adaptive to altitude because this is only bad when it's in the context of these. So how can we show you that that's functionally important? We take blood platelets from patients. We have a cell line that lacks its own mitochondria. We fuse the blood platelets, transferring the mitochondrial DNA from the individual. We select for these somatic cell hybrids or cybrids. And we assay for complex 1. So here are N lineages F1 and B. Remember, F is the diabetes one. And you can see when we have this C mutation, we get a 15 to 30% reduction in complex 1. So it's functionally important. But now look at the relationship between the T allele for F and the T allele for B. That difference is greater than this difference. So B and F not only are relevant to each other, but relevant to the new mutations. And finally, if we look on the M lineage, the C mutation is as good as the T mutation on B. So again, background is everything. Context is what matters with mitochondrial energetics. So we can then use these kinds of lineages to do a lot of association studies, we and many others now, and they've correlated with risks for Alzheimer's, Parkinson's, macular degeneration, ALS, migraine, psychiatric disorder, stroke, diabetes, cardiovascular disease, metabolic syndrome, sepsis, IgE, asthma, AIDS, osteoarthritis, aging, a whole bunch of cancers, athletic performance. Okay. All right. So how can I prove to you that, in fact, these are relevant to disease? So what we did is we created a mouse which has a specific mitochondrial DNA point mutation. We originally made this to look at whether we could show that these mutations, in fact, cause the disease. So in one mouse cell line, we isolated a mutant with a frame shift in the ND6 gene and a missense mutation in the CO1 gene. We take that cell, we remove its nucleus, take the cytoplasmic fragment, we created a female embryonic stem cell, fused in the cytoplasm, and then we took this mitochondrial-carrying embryonic stem cell, put it in a blastocyst, put it into a foster mother, got a chimeric female, and bred for the transmission of the dominant agouti locus through the female line, thus picking up the mutant mitochondrial DNA. Okay, so this shows, in fact, the mutation, which was an insertion of an extra C in the ND6 gene, throwing it out of frame. But this particular embryonic stem cell deleted the adjacent T, and these cells now have reverted back to wild-type ND6. They still all carry the missense mutation in the CO1. So after many tries, we finally got this particular embryonic stem cell line to go germline, and the female that had this line had in different organs different percentages of the mutant and normal mitochondrial DNA, replicative segregation. So we then took this female and we crossed her with nuclear match control. She started out with about 47% frame shift. Her first and second litters were all 16% frame shift. The third litter was 6%, fourth, fifth, and sixth were 0%. So there was a selective loss of the frame shift. We then took the 16% and mated them, and they gave 6 or 0, 6 or 0. We then superopulated the 16% and asked what their ovaries looked like. And their oocytes had up to 16%, but never more, always less. Now, this is really weird, because if this was stochastic, you would expect a Gaussian distribution. But in fact, this is a truncated distribution. What that means is inside the ovary of every mammalian female, there is a system that selects against the most deleterious mitochondrial DNA mutations and throws them out. That's a pre-fertilization selection. Why would that be important? Because if you have an exquisitely high mutation rate for genes that are absolutely critical for life, and you let all those mutations into the population would go extinct. So since mitochondria can function in a single cell, you can now have selective pressure on the cell before you ever get a zygote. And that allows us then to have a very high mutation rate without high genetic load, thus making this the intra-specific adaptive engine that I was just telling you about. Okay. Now, let's say we segregate the frame shift mutation. Now we have the CO1 mutation. It creates a 50% reduction in cytochrome oxidase, gives you degenerating muscle fibers with abnormal mitochondria, cardiomyopathy, and a whole bunch of neurological and muscle symptoms. Let's talk about another disease. This is a family that was originally reported by Massimo Zeviani. This woman had 50% of the mutation that changed this ND6 gene, proline codon 25, to leucine. And she had optic atrophy and cerebellar ataxia. Her sister had only 5% mutant and was perfectly phenotypically normal. But of all three of her consorts, all of their children had 100% mutant and all died. So here we have the stochastic inheritance of this heteroplasmic mutation, giving what would otherwise be an unintelligible phenotype. And I just received, this is an aside, I just received an email the other day, an article that just came out that argues that, in fact, Darwin's family, and the reason Darwin was sick is that he had the MELAS mutation. And if you trace his pedigree, everybody on the maternal lineage is affected with MELAS phenotypes. But anyway, that's an aside. So what we have then is this kind of random segregation of heteroplasmy. So how can we see if this is really the causal mutation? So we screened for many years to find a cell line that had exactly that mutation. Here it is, ND6 proline 25 to leucine. We enucleate that cell, we have a cytoplasmic fragment, we take our female embryonic stem cell, we put in the mutant mitochondrial DNA, put in the blastocyst, put it in the foster mother, and we transmit the mutation. So what do we get? So this is just looking at the optic nerve. This is the control optic nerve with the normal axonal fibers, and you can see in the mutant we have swollen fibers. And in fact, the small caliber, high energy fibers are preferentially affected. You can see that the red are the normal mice and the blue are the mutant, and you can see that there's a bias towards small caliber fibers in the wild type versus the blue, and that gets worse as they age. If we then look at these more generally, we see demyelination with highly abnormal mitochondria, which is what you would see in multiple sclerosis. And what's interesting is that in Leber's families, males go blind, but females get multiple sclerosis. Okay, now this is hard work of a number of our colleagues, which I'll introduce a little bit later, but this is just looking at this proline 25 leucine cell line. We see a decline in the reactivity for tyrosine hydroxylase in the basal ganglia. We see an increase in anxiety and a depressive effect. We see severe effects on motor, and we see a learning disability. And if we now do a MRI, we can see that we're getting specific alterations in the mossy fiber density. So this mitochondrial DNA point mutation, nucleus perfectly normal, is giving you all the clinical neurological phenotypes of what we would call Parkinson's disease. So that means that this must be an energetic disease, because that's the only change that has occurred. Okay, so how can we tell more about this disease? Well, now that we have a mouse, we could actually isolate the synaptosomes, and then we can study the mitochondria in situ. That's very important. So when we do that, we find that this complex 1 defect, which reduces energetics 30%, has no effect on the ATP level. But it does have a 30% reduction in respiration at rest, but as we increase the demand on energetics, the respiration rate can go up to normal. But what happens then is the reactive oxygen species production goes to very high levels. So we think that this disease is not about energetics, it's about reactive oxygen species toxicity. And we think that's an important factor, and some of the things we heard earlier today consistent with that. Now let's just age these CO1 and ND6 mice to 24 months. Now we see that we get insulin resistance. So here is the insulin level of the mutant versus the wild type for the ND6 and the CO1. And for the CO1, we also now have a glucose intolerance. So this point mutation then is generating all the phenotypes that we see in common diseases. So, okay, those are pathogenic mutations. But what about just naturally occurring variant like you all have? So we decided to try and mix two naturally occurring mitochondrial DNAs, one NZB from the mouse and one 129. We mixed NZB with the 129 mitochondrial DNA, put it into the animal. Now we have the founder female, and that's the NZB mitochondrial DNA, that's the 129. This is her daughter. We mated her, and all of her offspring, males or females, have the heteroplasmy, and the daughters transmit it, whereas the males do not. So it's maternally inherited. So now we have basically about 91 nucleotide differences, 15 amino acid, 5 tRNA, 7 ribosomal, and 11 control region. Now these animals have been back-crossed 20 generations onto a common inbred background. So there's no nuclear variation within what we can control for. So now what we do is we take the heteroplasmic animal and we let it segregate to pure 129, pure NZB, and heteroplasmic, and then we ask, what's the phenotype? So when we look at the phenotype, if we just look at their activity, we find that the 129 and the NZB are active at night, just as they should be. But the heteroplasmic animals are hypoactive. And in fact, they're hypoactive associated with food, decreased food intake. Their respiratory control ratio is slightly lower, but when we put them into a threatening environment, they become hyper-excitable. Now, if we ask, can they learn, here in black is the NZB, open circles 129, and blue is the heteroplasmic animals with different routes of learning. And the learning is to take this open field with all these holes around the middle and around the outside, and there's a black box there, and they're colored symbols so they can find their way, and they learn over time how to find the box and hide. So all three lines learn, although the heteroplasmic learns more slowly. But then Megan, who did this work, let the animals rest for a day and then asked them if they could remember the task. And the 129 and the NZB immediately ran and jumped in the hole. But the heteroplasmic animal acted like it had never seen that before. It completely forgotten. So what we can see here is by just having two perfectly normal mitochondrial DNAs, we can get, in fact, many of the features of learning disabilities and obsessive-compulsive behavior and depression. Okay, so now let's look at a nuclear mutation. This is the adenine nucleotide translocator that exchanges ATP and ADP, and this is a heart-muscle-brain isoform, because there are other isoforms that make this non-lethal. This mutation occurred in Switzerland about 500 years ago. We've traced this huge pedigree in North America, and ultimately we get consanguineous marriages where these people have heart disease. And that maps to chromosome 4, and it's a frame shift mutation by deletion of this A in the ANT gene. And so these people then have heart disease and myopathy. If we look at their echocardiogram, every heartbeat in the wild-type humans is very normal. But of the ANT, we see highly dysrhythmic effects. This is called velocity vector imaging. Green is normal, red is abnormal. Okay, but there's a major difference. Some of the individuals can live till they're 40 with just hypertrophic cardiomyopathy. But some of the individuals at very early childhood develop a massive dilated cardiomyopathy, require heart transplant. They have all kinds of pathology, abnormal mitochondria, and would go on and die. So why are there differences between the two types of individuals with exactly the same nuclear gene? And it's all about the mitochondrial haplogroup. Those with haplogroup H have perfectly normal, have hypertrophic cardiomyopathy that does not progress, but those with haplogroup U progress to transplantation. So to prove to you that that's true, we created a mouse with the same, with an ANT defect. Normal mouse will run. This is the oxygen use in red and the CO2 given off, and it will be using aerobic exercise and can run for very long periods of time. The ANT1 knockout mouse starts running and literally falls down. And it has then an accumulation of mitochondria by trichrome stain, cytochrome oxidase, succinate dehydrogenase, massive proliferation of mitochondria, and hypertrophic cardiomyopathy. Okay, so that mutation creates the cardiomyopathy. So now, this are then the heart echoes of wild type, CO1, ND6, ANT, ANT-CO1, and ANT-ND6. And note that this heart is now massively dilated and could hardly beat. So this is the exact phenotypic repertoire that we see with the family with H versus U mitochondrial DNA. Okay? So we can then quantify that by velocity vector imaging. We can see that in wild type we get uniform heartbeats. In the mutant we have dysrhythmia, and the velocity vector images show that same phenotype. Okay, so we can then now quantify all this. So now we're looking at different cardiac effects. We see that in this particular case, which is diastolic diameter of the left ventricle, that the wild-type CO1 and ND6 are pretty uniform. ANT is higher. ANT-CO1 is not too bad, but ANT-ND6 is very bad. This is ejection fraction. And if we now look at these velocity vector images, you can see that we, in fact, have a direct relationship between the mitochondrial DNA and the nucleus. And if we look at lifespan, the ones with the ND6 mutation die very quickly relative to the wild type. The CO1 are next intermediate, and the others are, in fact, more normal. And these then you can see a progression of abnormal mitochondria. So what that shows then is that mitochondrial variation, naturally occurring variation, has also having a very big effect on the penetrance of even classic Mendelian nuclear genes. Okay, so the last point to be made is this is the family with the 3243 mutation. I saw this family in the early 80s. This woman had lactic acidosis and growth retardation. These are all the children she had. We could examine them. These were not available. They all had lactic acidosis, growth retardation, progressive dementia, stroke-like episodes, hypertrophic cardiomyopathy, cardiac conduction defect, and all of these young men and women died in their late teens or early 20s of either heart failure or status epilepticus, and the oxidative muscle fibers were degenerating with abnormal mitochondria with Wolff, Parkinson, White conduction defects. So what we did is took the heteroplasmic cell line from this person, And we then took one of these cell lines without mitochondrial DNA, and we fused the cytoplasm of that cell to this cell, and then that created cells with different percentages of mutant normal mitochondria. And we then actually cloned out a whole series of lines that covered the full range of genotypes. Because the question is, why is it that this mutation in this family, which was 70% mutant, had cardiomyopathy and stroke-like episodes, whereas when 10 to 30% mutant gives type 1 or type 2 diabetes or autism? And this is really bizarre because this is supposed to be an autoimmune disease and this is supposed to be metabolic, and yet the same mutation causes both diseases. And yet at very high levels will kill you as an infant. So we could then ask, what's different about these different genotypes? So we can see as the percentage of mutant mitochondrial DNAs increases, the enzymes that have mitochondrial DNA-encoded subunits decline. You can see that as the percentage of mutant increases, the protein synthesis incorporation in this mitochondrial DNA-encoded subunit declines. Well, there is a threshold effect that as oxygen consumption stays relatively constant till over about 60% and then falls off. We see alterations in mitochondrial morphology, and most striking, we see that for the first from 0, 20 to 30%, a remarkable decline in cell size. So we're seeing then simple changes in mitochondrial genotype by only 10% has a big effect on cell physiology. Now, to cut a long story short, we've then done RNA-seq on all these cell lines and analyzed all of the transcriptional pathways. And what we see that in the 20 to 30% that's associated with type 1 and type 2 diabetes and, in fact, autism, that's one transcriptional profile from the nucleus. If we look 50 to 90%, which is the neurodegenerative disease, that's another transcriptional profile. And if we look at 100%, which kills the infants, that's yet a third. So if we do a principal component analysis, this is the normal gene expression profile. This is the 20 to 30% over here. Completely opposite is the 50 to 90%, and then 100%. So what this is saying is subtle changes in mitochondrial bioenergetics is causing phase shifts in the epigenome expression of the nuclear genes. And it's this epigenomic expression that we believe creates these phenotypic changes that are related to cancer, to metabolic syndrome, to diabetes, and so on. So it's the nuclear cytoplasmic crosstalk that's important. So the last point to be made is that we're really two organisms. The mitochondrial organism that generates most of the energy and has a quantitative genetics. The nuclear cytosolic organism that has a quantized genetics and anatomy and the anatomy of energetics. This organism has a very high mutation rate because it has this prefertilization selection, so it can tolerate a high mutation rate. This has a very low mutation rate because all these mutations have to go through development before they can be acted on by selection, and therefore a high mutation rate would be very deleterious. So in fact, most of the variation within the species is going to be in the mitochondrial DNA, not in the low mutation nuclear DNA. Finally, the energetics of the mitochondria make the ATP, the S-adenosylmethionine, and the acetyl-CoA that in fact regulate the signal transduction pathways and the epigenome. So why would that be? Because the nucleus, remember, is energy limited. It can't do anything without enough energy. So it has to know what the energy flux is through the mitochondria. So why did the epigenome evolve? It evolved by using mitochondrial high-energy intermediates to change the expression of the nuclear genes so that they would only be replicated, transcribed, and translated when there was enough energy. So now that says that if this concept is correct, then these are the major intraspecific variables that will relate to phenotype, and this is primarily intraspecific. So I'd like to end then by just thanking many years of wonderful colleagues, and I don't even know that that's all that clear, but people that are here are Alicia, Danielle, Kirsten, Piotr, Megan, Martin, Caitlin, and Jesus, all very important members of our group. People that have made major contributions are Wei Wei Fan, Brian Lin, Grant McGregor, Ryan Morrow, Mark Shapley, and Katrina Waymeyer. Our studies in Tibet were done with the people at 3rd Military Medical University in Chongqing with Fuyan Ji. The ophthalmological studies with Alfredo Sedone and Valerio Corelli. Our epigenomic studies with my long-term friend Paolo Sassoni Corsi. The cardiology studies with Jargat Narula. The family that we studied in Pennsylvania with Kevin Strauss of the Clinic for Special Children, and then our long-term anthropological studies, and thank you very much.
Terrific. We have time for a few questions. The microphones are in the aisles. Please use them so people watching on the video can hear the question, and we are open for those questions. While they're thinking about it, you showed that data about NZB in 129, where the heteroplasmy was the problem, and the homoplasmy of either type was fine. Is this explicable on the basis of some multimer problem that happens when you have both of those present in the same cell?
Yes. So thank you, Francis. So again, let me reiterate, these two mitochondrial DNAs are perfectly normal mice where they're just collected from different environments. But they still differ by about as many nucleotides as you all differ in. But some of those are amino acid substitutions, because remember, anything that's in a different environment will have a slightly different amino acid substitution. So now what we've done is taken one mitochondrial DNA with, say, 2 or 3 functional amino acid changes, and another mitochondrial DNA with 3 or 4 different ones. We've now put them together and asked them to make the same multi-subunit complexes. And so now you have a problem of a uniform nucleus, but trying to insert different polypeptides into that site.
So when you have cells that have some mitochondria with mitochondrial DNA mutations and other mitochondria without the mutations, have you tried to manipulate the cells to, for example, enhance mitophagy and try and rid the cells of the mutant mitochondria? And would you predict manipulations done in vivo? So, for example, like maybe intermittent fasting or something which will inhibit mTOR and upregulate autophagy would have a benefit in any of these patients?
Absolutely. Very, very good question, Mark. So I didn't have time, obviously, for obvious reasons, to go into our efforts to develop new diagnostics and new therapeutics. So this is in the therapeutic area. I would like to mention Carlos Moraes' work. What he's done is he has created enzymes that will see specific mitochondrial DNA mutations, let's say a pathogenic one, then put that enzyme in the nucleus with a mitochondrial targeting peptide, and then that protein goes into the mitochondria, sees that mitochondrial DNA as foreign, and digests it. So he can, in fact, change the heteroplasmy by that directed problem. We still have a problem how to deliver such a vector to all the cells in the body, but we're actively working on adeno-associated viral ways of doing those kinds of things. Now, Eric Schoen has taken cells that are heteroplasmic with pathogenic mutations, and he's put them into a ketogenic diet, which is basically burning fat, and he can shift the heteroplasmy in that way. And that might be a more credible way of treating patients. But again, we don't have the sample size to do those kinds of clinical trial studies at this point. The National Institute of Child Health has been supporting a major effort to begin to get a patient registry so that we can do clinical trials, and we're very grateful for their support on that.
Great. Over here.
Thank you very much, Professor Wallace, for your presentation as well as your seminal contributions in this field. I'm Evangel Fan from Dr. Bott's group at NIA in Baltimore. So I think I have the same question as I asked you last year. It is how to give an exact definition of mitochondrial disease or not. For example, some of the diseases based on the mutation of the mitochondrial encoded proteins or the mitochondrial localized proteins, we can see it's a definite of mitochondrial disease. But for some other diseases, such as a mutation of the other proteins in not in the mitochondria, but it can affect the mitochondrial function as well as the mitochondrial activity. So how do you think those disorders belong to mitochondrial disease or other disease with mitochondrial phenotype? Thank you very much.
No, please say your name more clearly.
Evangel Fan.
Okay,
Yes.
and this gentleman is working with Will Bohr at NIA, and they are doing, I think, a very important study. They are trying to use information sciences to go through the literature and define the phenotypes of known mitochondrial disease to try and develop algorithm for better diagnosis. This is an area of discussion in the field. There are some people that would like to split the field of mitochondrial medicine into what they call primary disease and secondary disease. I actually feel that this is really more of a continuum, and that there are diseases that are due to mitochondrial DNA mutations, there are diseases due to nuclear mutations in mitochondrial genes, but there are also the interactions of these genes and of anatomical nuclear genes as well. So, at this particular point, we have a big problem, because what we see is different mitochondrial DNA mutations or different mutations in nuclear genes. They can give quite radically different phenotypes. That is, I think, one of the most important questions, and why we spent so many years trying to make mouse models, so we could actually try and look at one mutation, define the phenotype. But why would there be such phenotypic variation? Why is the brain affected in some individuals' heart and the others' eye and the others' kidney and the others? And I think that's because the mitochondria is so pivotal to so many functions in the body. And therefore, you can perturb it in many different ways. You could have too much ROS, you could have a redox change, you could have calcium deregulation, you can have energetics, you could have changes in metabolites, in fact, all of which we've heard earlier today. And those are going to affect organ systems in different ways. And so I think what has happened is we want everything to be linear, but I think this is such a pivotal system that it affects every organ system, every institute here at NIH, and will affect them in different ways, and we can only understand that by truly understanding the physiology of this organelle and its relation to the nucleus. And I don't that much help, but that's where we're at right now.
Great. We're a little over time, but we'll take 2 more questions here.
All right, thank you very much. Very interesting talk. So I was wondering about the connection between mitochondria and aging. And so in mitochondrial diseases, you need sometimes 70% clonal expansion of the mutation to get a phenotype. Whereas in aging, so it's been postulated that, or proposed that with aging you get accumulation of DNA damage that leads to mitochondrial dysfunction and so. But the accumulations that you see in aging is much lower frequency. So how do you, is that something that you could elaborate on? Or?
Right. So that's a really good question. But remember, the cell has 5,000 targets. And so the concept that we have from, and being a Mendelian geneticist, the concept we have, you have 2 alleles. You get 1 allele, 2 allele mutant, you're done. But here we could have 1 out of 5,000 that are mutant. So you could have 5,000 mutants, a different one in every mitochondrial DNA. And that concept is also very relevant in cancer. We actually are working with mitochondrial heteroplasmy in cancer and how it affects cancer cell growth. And I think that's a really big area. But how do you quantify that when you have many different mutations? So one of the things we spent a lot of time and money on is trying to develop ways to literally clonally analyze every single mitochondrial DNA, define its mutations, and then add that up as a population biology. And then you'll see that, in fact, even though any one mitochondrial mutation is very low, the sum total is actually quite prodigious. And we heard earlier today the studies by Niels Larsen and Prokhoff, where they've put in a mutator nuclear-encoded mitochondrial DNA polymerase, and that causes the animals to age prematurely. They don't get any one mutation. In fact, they get many, many different mutations. And again, you have to quantify every mitochondrial DNA separately. Okay.
Thanks.
Will have to be the last question, but I'm sure you can answer other questions in the library in a minute. Final question over here, yes.
You showed in your mouse studies that the deleterious alleles were progressively less in every generation, but yet in the human pedigrees, that doesn't happen. Can you speculate on why?
Yes, thanks. That's an excellent question. I perhaps didn't make that as clear as I should have. Our work at the current state shows that so in humans, the female generates about 3 to 5 million proto-oocytes. And then she will only ovulate about 400 viable eggs. So what's all the rest of those cells? Where are they going? What it seems is every time a female goes through the estrous cycle, many hundreds, if not 1,000, of these protooocytes begin to mature. And then, in fact, they are in competition with each other. And then almost all of them will die by apoptosis. And only the few will form a mature follicle and apoptose. So we've been very interested in why that is. And it turns out that the ones that have the highest oxidized glutathione are the ones that undergo apoptosis. So the idea is that the cells that have the most mutant mitochondria have the most oxidative stress. They oxidize glutathione, and that trips the permeability transition pore. So you have an intra-ovarian competition. Now, that competition is not on or off. So evolution wants variability to go into the species population. That's why I'm arguing it's the evolutionary radiation tool. So the threshold is not put at 0. It's right, it's up at a level. So we get a lot of 3243 mutation patients. That's pretty deleterious when it's homoplasmic, okay? But when it's at a low-level mutant, it gives you migraine headaches. So that's at the level probably where it's still not enough for the ovary to clean it out, but it's still enough for us to see the patients in the clinic. And I think that that's why we have preferentially so many specific mitochondrial mutations.
Well, please join the conversation in the library over coffee and cookies, but please thank our speaker again.